Assalamu alaikum wa rahmatullahi wa barakatuh. This is Elshimaa from Biotechnology Study Program. I will explain about glucose-based mixed culture fermentation in our microbial physiology course. I'm going to use this map, which is a colorful diagram presenting very different metabolic products, which are all important in industry. This diagram is actually a map showing how different microbes from different species in the same environment ferment glucose into different metabolic products. At the center of the map there is pyruvate, which is the main intermediate molecule that comes from glucose. Around pyruvate there are the different six sections, each represent a different metabolic role. Section 1 in the red-brown color starts from glucose, then glyceraldehyde 3P, G3P, then PEP, and then pyruvate. This is the main EMP pathway, and almost all microbes can perform this pathway, especially under anaerobic conditions. Around pyruvate there are different sections, each represent a metabolic route. Sections from 2, 3, 4, 5, 6 all represent a group or a set of possible final products, each with a superscript that represent the redox state. R means reduced, N means neutral, and O means oxidized. This is very important because fermentation is not only making ATP, it is also keeping the balance of NADH and NAD ions stable. For example, if the cell has too much NADH, it will produce reduced products like ethanol, butanol, and acetone or propanol. If the cell has too little NADH, it will produce oxidized products like formate, succinate, and this. The possible products are produced depending on the environment condition, enzyme availability, and redox needs, and the species. And this network is very important in almost all fermentation industries. Do microbes use EMP or ED or both pathways in fermentation? Generally, EMP or Embdemeyer-Hoff-Parnas pathway is preferred. EMP use is clearly shown in section 1 in the map. It is preferred or dominates because it yields more ATP and more NADH, and most fermentative microbes like E. coli, yeast, and lactic acid bacteria, they naturally depend on or rely on EMP pathway. ED pathway is used by fewer organisms like Zymomonas mobilis and some Pseudomonas. It produces only one ATP molecule, which is not ideal for fermentation environments, which are usually anaerobic conditions. So in general, EMP is preferred as shown in the original map, but ED may occur because in mixed culture, some microbes that use ED may present, but they contribute very little to the general fermentation. Now, what if the substrate change mid-fermentation process? Naturally, this may occur because the original substrate depleted or microbes consume intermediate metabolites or a secondary carbon source becomes available. So what if the substrate changes? Many things may change also. The first thing is carbon catabolite repression system changes. Microbes rebuild their priority and change their enzyme into the new substrate available. For example, switching from glucose to glycerol requires activation of specific enzymes, which may require time. 2. Rerouting of metabolism or the metabolic flux changing. All arrows in section 2, 3, and 4, and 5, and 6, they readjust. If the new substrate is more reduced, like glycerol, microbes will produce more reduced products like ethanol, butanediol, or butanol. And if the new substrate is more oxidized, microbes will produce more oxidized products like acetate, succinate, or other oxidized products. 3. Redox change or rebalancing. Each substrate has a specific redox load. Microbes immediately adjust end products to rebalance the ions from NADH and NAD ion. And finally, a temporary instability. Microbes do not switch instantly, they undergo a lag phase, transforming from the first enzymes to the second enzymes for the new substrate. So if the substrate changes, microbes adapt to the new substrate and fermentation does not stop. They change their metabolism, they change their enzymes, they redirect their product pathways and shift to new redox pathways. Now, as I said, mixed culture fermentation is very important in industry, and each area of the diagram has many influences on the industry. For example, the first section, section 2, from G3P to D-lactate or glycerol or 1,3-propanediol is very important in industry. Glycerol is used in cosmetics, pharmaceuticals, and many chemical industries. Propanediol, 1,3-propanediol is a polymer used in polymer manufacturing like fiber production. D-lactate is very important also. It's used in biodegradable plastics. This branch is has very heavy products in fermentation. Area 3 is essential in many traditional and modern fermentation industries. It starts from pyruvate to acetone, ethanol, and 2,3-butanediol. Acetone is used as a food flavoring agent and in fragrances. 2,3-butanediol is used in rubber production, is an antifreeze agent, and as a solvent. Ethanol is also a very important product from fermentation. It's used as a biofuel, beverage, and sanitizers. Area 4 is a very heavy area in fermentation industry. It starts from pyruvate and it has many products from formate, acetyl-CoA, which produce acetone, acetate, butyrate, and butanol. Butanol is used as a solvent. Butyrate can be used in flavoring agents. Acetate is the vinegar is important in vinegar production. Acetone has many applications in industry also. Area 5 is also very important in industry. It starts from L-lactic acid to propionate and propanol. L-lactic acid is a backbone for PLA bioplastic. Propionate is used in food preservative. It is an antifungal in bread products. Propanol is used as a solvent in chemical industries. And the last area 6, has many influences in industry, starting from decarboxylic acid to propionate or succinate. Propionate is a food preservative, especially in bread. Succinate is a very important chemical in pharmaceuticals, in solvents, and in bioplastic production. Generally, all branches are important for industry, and industries choose specific microbes to push carbon flux into the desired product. And these are the references I used to present this presentation. And finally, thank you for your attention and your listening.